And today, my talk is about heterogeneous nature of antigen-presenting cells in the peritoneal cavity. Immune system breaks into two main branches, innate and adaptive immunity. Innate immunity is non-specific first defense mechanism, which occurs rapidly right after antigen exposure. Adaptive immunity is specific response, which also can produce memory responses. Unlike innate responses, adaptive immunity cannot act directly after antigen exposure, and stimulation by an antigen-presenting cell is prerequisite process to achieve a functional adaptive immune responses. Dendritic cells, which are the main topic of this talk, are the most potent antigen-presenting cells that provides the essential bridge between innate and adaptive immunity. The main function of dendritic cell is inducing adequate T-cell responses. Dendritic cells use three signals to modulate various types of immune responses: MHC peptide complex, co-stimulatory molecules, and cytokines. Combination of these three signals lead to various types of immune responses. They induce specific type of immune responses against respective antigens, whereas inducing immune tolerance against self-antigens and common cells. They also imprint homing receptors on T cells and induce formation of memory cells. Dendritic cells are a heterogeneous group of cells consisted of various subsets. And with recent advancement in cell analysis, dendritic cells are constantly being divided into smaller subsets with specific locations and functions. Not only dendritic cells, but macrophages are also being split up into more detailed subsets, varying tissue, function, and origin. Classically in immunology, dendritic cells were described by their distinctive surface markers CD11C and MHC2, and macrophages were described as cells expressing FO80 and CD115 and other macrophage markers. However, detailed studies and technology reveal the complexity of the surface markers of myeloid cells. In some tissues, subset of dendritic cells and macrophages share their markers, and also they share unique functions that used to differentiate them. Therefore, to accurately distinguish these cells, it became more important to assess various factors of each tissue and apply more specific markers to identify one another. The peritoneal cavity is a specialized compartment between the parietal and visceral peritoneum. Along with many organs, various immune cells reside in the peritoneal cavity and induce immune responses. Mononuclear phagocytes, mainly macrophages, take up half of the immune cell in the peritoneal cavity. There are two types of macrophages, which express high levels of FO80 and low-level MHC2, large peritoneal macrophages, and small peritoneal macrophages, which express low level of FO80 and high level of MHC2. As mentioned, MHC2 is a commonly used marker to distinguish dendritic cell as well. Even with the sharing markers between SPMs and dendritic cells, there was no definite description to distinguish small peritoneal macrophages and peritoneal dendritic cells, which made it confusing to clearly gauge dendritic cell in early years of my research as well. Recently, there are reports on cells expressing both 11C and CD115, which are the commonly used marker for dendritic cells and macrophages. And these ambiguous cells have not been described yet.